Well, welcome to Chapter 21, Bio 121. We're looking now at viruses, and one thing that I want to point out to you that this textbook is not doing, that I want you to consider doing is, sure, these are structures that are not capable of living on their own. And as your textbook describes, they are the perfect, what we consider to be parasite. They require everything in the host cell that they are infecting to do what they need to do next. But one thing I want you to consider is what has been their role in causing or driving any form of reproduction inside of humans or organisms on the face of the planet, because you're going to see viruses essentially can infect every single organism on the face of the planet. How have they driven what we consider to be evolution, and why aren't you taught to think about what this might actually be? So we're going to start with basic virus structure and, what they may do, what they may look like, how we classify them, and the major tools that were required for us to identify them. But you already know them. It's an electron microscope. And then ultimately, I want to finish this off with, if this is a virus and it's encapsulated, what are these things we call prions, and what are they doing at the end? And why could it be happening here? And why do we actually care? Okay. There's some really good reasons why you should care, not just because it's going to be on your final exam. Okay, so viruses are different from living cells. They are acellular, they have no metabolism and cannot grow. They depend on host cells to make copies of themselves. We have no idea where they actually all came from, none whatsoever. We have ideas as how they may have evolved over time amongst themselves, but where it actually came from, nobody knows, or where they've all come from, nobody knows. They have collected pieces of nucleic acids from various sources and created a hodgepodge. And I think that's probably the coolest thing about them, right? They have, if we think about things as being energy requiring, they understand where to find energy, where to get the energy using the most minimal amount of information. And then they can essentially infect nearly all life forms. And that doesn't mean any virus can infect any life form. You're going to see that unique viruses infect unique life forms. Okay. Not only will they be species specific, but they're also tissue specific inside of the fact of where they are. And even though you're sick and tired of thinking about coronaviruses, and heavenly knows so am I, the idea that they target a specific receptor that's part of your emergency response system to changes in water fluid inside of your body by attaching the ACE receptors, right? It means that they have figured out that that is a level of vulnerability excuse me, in your body, and in doing so, they figured out how to co-opt it for their own need, meaning you need that receptor, which means those viruses have figured out you need this, therefore that's not going to change. That's some level of stasis for them to move into you as a living organism. Now, viruses were known to exist before they were ever seen. The first examples are tobacco Mosaic virus, where essentially you would ruin thousands and thousands of crops worth of tobacco, which back in the day was the money-making crop. It's driven lots of society as we know it. But you would see the change in the leaf and then there would go your crop, and ultimately people would find, if this is 50 nanometers here, they would find that this was the material that was infecting those leaves. We've seen these guys before too. This is Bacteriophage T4, 1930s. Remember, we did our S35 and P32 experiment demonstrating that the P32 on the capsid stayed outside of the cells, and the P32 and the inside of the cells went inside with the nucleic acids, and the S35 that was part of the capsid structures stayed outside, which is pretty cool when you think about how they were doing or what they were doing. Notice what they're doing here, of course, is this virus is actually infecting a prokaryotic cell. And you'll see that when you figure out how specific each of these viruses can be, we can take advantage of this. And a side note, that genome that I think I had you guys look up before cutting with PST EcoR1, and I don't remember what the other endonuclease is, that's the genome that's found inside of this organism here. Okay? Whoops. No worries, just trying to record here.